Assalamualaikum and hello everyone. Today we're going to talk about methods in gene cloning. Basically, there are 5 steps in gene cloning. The first step is isolate. The second one is cleave or cut. The third one is insertion. The fourth one is transformation and amplification. And the last one is screening. In our first step, we're going to isolate plasmid from bacteria cell. And we're also going to isolate target gene from our DNA source, which is human cell. And typically, we're going to use E. coli as our plasmid. Our second step is cleave or cut DNA. Firstly, we're going to add restriction enzyme into the plasmid and target DNA. Our restriction enzyme for both plasmid and target DNA must be same and compatible so that we're going to have complementary sticky end. Complementary end is needed so that we can move forward to our third step. Our third step is insertion process. In this step, we're going to add plasmid, target DNA, and DNA ligase together. Why did we need to have DNA ligase? This is because we're going to need phosphodiester bond to combine DNA fragment and plasmid DNA together so that we're going to have recombinant DNA. Move forward to our next step, which is transformation and amplification. In this step, we're going to perform transformation process by introduce or transfer plasmid into the host cell. Some cell may take up non-recombinant plasmid, recombinant plasmid, and some of them may not have plasmid at all. First of all, we're going to treat the bacteria, which is the host cell, with calcium ion and a brief of heat shock. This is because we're going to make a hole appears in the cell surface membrane so that they will become permeable and allow DNA and plasmid to enter. Next, we're going to culture the bacteria on nutrient-containing agar medium supplemented with ampicillin and X-cal, which is a molecule that resembling a lactose. Next, we're going to incubate until colonies grows, and only a cell that took up a plasmid which has the MPR gene will reproduce and form a colony. And the last step is screening. In screening process, we're going to have 3 types of screening, which is antibiotic screening, blue-white screening, and the lastly is nucleic acid probe hybridization. In antibiotic screening, we're going to choose clones with a vector antibiotic resistant because only bacteria that have the ampicillin-resistant plasmid will grow in the antibiotic, while the bacteria that do not have the ampicillin-resistant plasmid will not grow. The second type of screening is blue-white screening. In blue-white screening, we're going to differentiate non-recombinant plasmid and recombinant plasmid. Non-recombinant plasmid can intake Laczac that encode beta-galactosidase so they can hydrolyze H-gel and their colonies will be blue. While recombinant plasmid, their Laczac is disrupted and they cannot encode beta-galactosidase, so they cannot hydrolyze H-gel and therefore their colonies will be white. And lastly, we're going to have nucleic acid probe hybridization. Firstly, we're going to have 1,000 of recombinant DNA molecules in bacterial cell. They are screened to find the specific DNA sequence of interest. Then, we're going to have a common approach to detect a specific DNA sequence involved using nucleic acid probe. So, that's all. Thank you. Okay, that's all. Thank you.